Hello students, this is Vedant Mirza. Today we are discussing genetics review. In genetics, we discuss cross and how the different alleles combinations can come across in inherited patterns. So, for example, hemoglobin, the beta chain is coded for by two different alleles: the normal trait, which is HB capital A, or the sickle cell trait, which is HB capital S. You can see there's a point mutation which is occurred at a certain point, changing the nucleotide from T to A. So a person could be normal capital HB, HB capital A, HB capital A. The person could be carrier for sickle cell trait or having sickle cell anemia. These three subjects are present in our environment. If you just perform a Punnett square for HBA, HBS, and HBA, if you cross them together, so you will get a result here. When we are using Punnett square, we try to place the female gametes at the top and the male gametes by the side. The gametes here, HBA, HBF are male. You know that the alleles will be separating out during meiosis, so HBA will separate and the other HBA will be in the other gamete. Similarly, in female with HBA, HBS, one HBA will be in one gamete, the other HBS will be in the other gamete. So you just cross these together to see the outcome. And you can see the outcome that the normal hemoglobin HbA will be present in 50% individuals, and sickle cell will be present in 50. Sickle cell carrier will be present in the 50% individual as well. So there will be 50% chances of both. If two carriers are crossed, for example, HbA, HbS, and HbA, HbS, male and female are crossed together, you can see that HbA, HbS will be crossed together. So a normal male, a normal individual with normal hemoglobin and sickle cell carrier, and once will the sickle cell anemia will be reduced here. So 1:2:1 will be your ratio here. So there's 25% chance of normal male, 25% chance of a sickle cell individual, not just male, normal male or female either, because it's not a sex-linked trait. So the person will be normal. The person 25% chance that the person could be having sickle cell anemia, or 50% chance that the individual will be carrier for this trait. What need to be kept in mind is that if the parents are the carriers of the same recessive or autosomal recessive trait, so there will be 1 in 4 chance that the offspring will have the trait, or 25% chance of the offspring suffering from the disease, which you can see in this cross as well. If we take one more example of crossing a red flower with a white flower, and we know that red and white flower both are dominant features, so red and white flower when crossed together will produce F1 generation, will be all where all the flowers will be pink, they all be CRCW. And when the CRCW flowers F1 generation are crossed together, you can see the initial ratio being regenerated. So you can just perform this cross and see the outcome. Why do we perform test cross? Because test cross is used for, if for example, if an organism is showing the dominant trait in the phenotype with the organism who is homozygous recessive for the same trait, you can just cross them together and you can see whether the dominant individual was a homozygous dominant or the heterozygous dominant. For example, a guinea pig with a coat color black could be crossed with a coat color white, and you can see whether the organism was capital B, or capital B, small b. So if you cross this black guinea pig with the white guinea pig, you can see if the 100% offsprings are black, that means that the offspring in question, the organism in question initially was almost like a dominant capital B, capital B. But if 50% black and 50% white are produced, that means that the organism that you are discussing was with the dominant black coat was actually a carrier and was having a recessive allele for white coat color as well, but that was masked by the presence of the dominant allele capital B. Similarly, if you can perform a crisscross between a purple stem tomato plant, a green stem tomato plant, one having capital A, one having the small a, they both are purebred parents. Purebred means they are homozygous. One is homozygous dominant, the other is homozygous recessive. The offsprings of the F1 generation will all be purple. They will be all hybrids. Now, if you cross this F1 offspring with the recessive of the same features, that is capital A, small a with a small a, you will get 50% capital A, small a, and 50% will be small a, small a, the same thing that I just discussed with the black guinea pig. That's it for now. Thank you.